So welcome back for this afternoon session. Really nice to see so many people staying through the whole day. So thank you for that. I want to give a huge shout out to the apparently 29 people who are online. Maybe when we get to a break, I'll quickly read out their names if you wonder who's there. And without further ado, we'll move on to our first speaker, who's Matt Dietrich, who's going to talk to us about machine learning. So over to you, Matt. Oh, and you need to speak.
All right. Hello, everybody. Like Hillary said, I'm Matthew Dietrich. I'm a data manager here at Archbold Biological Station. And my goal is to talk a little bit about some big technology buzzwords like artificial intelligence, machine learning, big data, and put them into context with what we're doing here at the Predator Prey Lab, specifically with the Florida Corridor Observatory project. So, main question, what is machine learning? First, we have to step back and kind of put it into a larger context, specifically kind of this fundamental basis of computer science. And so this will include things like our mathematical operations, conditionals loops, and basically fundamental programming operations. Some ways that we use that here at Archbold is our field sensor function and our data flows. And then if we try to move one step further, a little bit more complicated, we're getting into artificial intelligence. And so this, at its most basic, can be rule or logic-based decision-making. So some examples of this would be search algorithms or genetic algorithms, or just general optimization problems. And so how we use this here at Archbold is our auto-flagging of our weather data. So basically there's rules that are applied that show us whether or not we need to go back and manually review some of our data. And then if we keep stepping further, we're diving now into the realm of machine learning. And this can include linear regressions, decision trees, basically anything that uses training data to inform a model. And so this includes kind of a common example like your email spam filter, but also specifically what we're doing here at Archbold. All of our scientific output is models based off of data. And so if we go kind of this final deep learning level, that will be where we're getting into artificial neural networks like the Florida Corridor Observatory, and how we're trying to kind of auto ID images and audio as they come in. This is also now the realm of big data. You need very large data sets to train a reasonably accurate model with any sort of proficiency in the task that you're trying to get it to learn. So stepping back again, trying to think of typical programming, we're going to start from some initial state, perhaps like the ingredients in your kitchen. We're going to move forward through a series of specific operations and apply those in a predictable order. That would be like following or creating a recipe. And then we're going to try to have some sort of final result. Ideally, a cake. When you're programming things, oftentimes stuff is going to go wrong, and you're going to have to trial and error things, or things didn't get installed correctly. Any number of issues can come up, and those will have to be dealt with. But it's all kind of sequential in this way. So how is it different in machine learning with artificial neural networks? If we go back and think about baking a cake, instead of kind of trying to build an individual recipe or individual steps, this would be like if I just told you whether or not you baked a cake correctly 1,000,000 times. And so basically you have to reach the output goal many times, and you have to have some way to assess whether it was reached correctly or reached incorrectly. And then also the intermediate steps are difficult or impossible to specify. So we're starting to get to the point where in kind of traditional typical programming where you're following a recipe or creating a recipe, you can kind of say there's a cause and effect for each step that you're following along the way. When you're learning based off of correct or incorrect examples, those intermediate steps blend together in kind of unique ways that you can't always separate out. So another example, we could try to define something like a water bottle. If we wanted to do it a typical programming way, we'd define it with rules. We could say that it is generally cylindrical. It's used for some sort of purpose. It has a cap or cover, and it's made of a subset of materials. You could make this as specific as you need to get it to actually refer to the things that you're trying to refer to. But if we wanted to do this differently, we could just say, this is a water bottle. And these are water bottles. And all of these are also water bottles. And so that's more of what we're trying to do with artificial neural networks. So what are some basics that go into making an artificial neural network? I'm going to keep this at a pretty high level. There are a lot of tools that are available to make this pretty easy to set up. And basically, if you have these three pieces, or at least a general idea of these three pieces, it'll give you enough to be able to learn more or continue talking to me in the future. So the three main pieces that I'm going to talk about right now are data, and the model architecture, and the loss function, which is sometimes called the objective function. So first and foremost, you need a large amount of data, and most of that is going to be going into your training, but some of it you need to separate out for testing, basically validating that the model is doing what you want it to on things that you haven't trained it to accurately identify already. And then the model architecture, this is kind of the piece that gets very complicated, and a general overview of what that might look like is on the right. Mainly, we're focusing on the number of nodes or artificial neurons, and this can range anywhere from about tens of thousands to hundreds of billions. And just kind of to put that in perspective, the smaller model architectures at the tens of thousand range would be more or less equivalent to the biological neurons in like a slug. The models that we're using here in the Florida Corridor Observatory are on the range of tens to hundreds of millions of nodes. And that would be more or less like a mouse or other small mammals. And then the hundreds of billions is your kind of state-of-the-art ChatGPTs or self-driving cars. And that would be beyond the size of an African elephant or so. And then the loss function is basically going back to kind of the baking a cake example. This is that person that tells you whether or not you baked the cake correctly and how you kind of update yourself based on that information. So what does Corridor Observatory data look like? We are collecting image data, you can see that on the left, and audio data, you can see that on the right. It's hard in some ways to visualize the audio data. I would play a sound clip as well. But more or less, this is seen in the same way in both cases by the computer. So each pixel in both of these images is a series of values, either red, green, blue, and an intensity for the image, or a frequency, time, and intensity for the audio. And that will all go through as input to your model architecture. And then you will get some sort of output feature class. So kind of in the earlier diagram, this was just one orange node. But if we were trying to say, if we had 10 potentially ID'd species, we could say there is some confidence level for each species, and then each row would be a different input image that we fed through the model. So kind of the specific tool that we have been using right now for image ID is called Camera Trap Detector. And it initially came out of this paper by Michael Tabak and many others. And we are currently working with Amira Burns at USDA to kind of continue updating the models. And so we've used heavily version 2, and now we're just starting to see version 3 come out and starting to use and kind of test the results that we're seeing there as well. And so what went into version 2 was a lot of training images from Buck Island Ranch. Additional images came from other ranches nationwide. And so the model became particularly strong with ranch animals. It wasn't so great on things that were caught in scrub background, so images from cameras that were set out in the scrub. And it processed about 12 images per minute, 5 or 6 seconds an image, which is nice because you can leave it running overnight or as long as you need to. But with version 3, there was an updated model architecture, which made it significantly faster. We're processing about 500 images per minute. We've seen an increase in accuracy, and some data augmentation techniques were applied. So this leads to the increase in accuracy. Basically, if you have an image of a deer, normally that would just be one training image that you say this is a deer, and you can train on that. But you could also mirror it. You could stretch it in some ways. You could kind of fudge a little bit of the pixels here and there as long as it's still recognizably a deer. And now your single training image is 4 or more. So that makes your training data set go further. So here are kind of some very preliminary results of analysis between version 2 and version 3. So basically, we're looking at accuracy rate. On the left, we want this to be very high. Ideally, we're getting up to like 95% accuracy, or at least reaching human level accuracy rates. And then the confidence threshold at the bottom is basically what confidence from the output of the model are we allowing when we're looking at results. So as we raise the confidence threshold, we're dropping off a lot of the data set that we're using. But in a lot of cases, that's okay because we're setting out a lot of cameras and we're taking a lot of images and data points. So specifically, this is deer from Archbold Biological Station camera traps. And so these are some of the kind of testing data sets that we've set aside and haven't gone into training the model. And this is another example of bobcats at Archbold Biological Station. So we can see that in version 2, it kind of peaked at just under 75% accuracy at a 95 or 90 or so confidence threshold. And then in version 3, we're reaching more of like a low 90s% accuracy, 90% confidence threshold. So there are plenty of things to keep in mind. They come in kind of 2 categories. The first is how we're using this data currently, or how we're using this model currently. So like I said, a reasonably accurate model will allow us to drop the low confidence data and just stick with the very high confidence classifications. So for example, we're getting around 2 million images per year with what we have set up now. Depends on what animals are moving in front of the camera specifically. So if 60 to 80% of that is at this 95% accuracy or human level accuracy, then we'd be reaching around 1.2 to 1.6 million usable classifications per year. A quick note is that the camera traps take bursts of 10 images at a time. That would leave us with around 120 to 160,000 usable classification IDs. The second and third pieces all go into continuing to update the model going forward. So a bias in training is going to lead to a bias in results. We want to keep being able to identify where the model is weak and why. Typically that comes from the training data. A model can only be as good as its training data. Usually it's worse. So we have a lot of work cut out for us to keep updating this going forward, to keep providing the increased training data sets and still accurately classifying things ourselves as well. We don't want to send this all over to the machines to take over. With that, a lot of people are involved in this project. Thank you to everybody. And if I have time, I'd like to take a couple of questions.
At the back, Joe. Wait a minute, can you use the speaker so they don't mind picking from here? Thank you.
Hi, sorry. Can you increase the number of features by looking at the databases from various museum specimens, and that taking their pictures, and then increasing the number of features that you can look at, so then you can identify more organisms?
So yeah, I guess, so what I showed before for the feature class was just an example of feature class. So what's going into the model is basically features of everything in the image. We are actually playing around with additional metadata from the images, but I don't think it's set up for that right now.
So what's from online?
Emily Online asked, what are your thoughts on using AI rendered imagery to train models?
That I think is a very bad idea because basically the model is only as good as its training data set. So its output is only going to be at a certain level. And then if you train another model based on that, model is going to be slightly worse. I think there were actually a couple of research papers that have come out that after like, I don't know, 20 or so iterations of this, you just have garbage mess, basically. So
Yeah, go.
I was wondering if you could go back to the 2 graphs between version 2 and version 3 that you showed. And I just wanted to sort of if you could help me walk through, no, the one before this.
Okay.
Why did version 2 have bad accuracy at low confidence threshold versus, and then it gets better? Like, shouldn't it?
Yeah. So this low confidence threshold would mean that even if the model is like, say, 10% confident, that this should be labeled as deer or rodent of some sort or something. So basically, at low confidence, you'd expect to see more incorrect classifications. What's actually interesting here is that at high confidence, we're seeing this kind of steep. And just, I think that's two parts. And the first is that at very high confidences, you have a very small amount of actual labels that you're getting at confidence. So this is maybe like 5 or so different IDs level. And then another that we've seen is that the model version 2 and version 3 are always very, very confident at identifying images that have no animals in them. So it puts those as like 95 or 100% confidence, even if it shouldn't be.
It's a great answer. Thank you.
Is there a version 4 in the works?
Version 3 is currently like cutting edge for camera trap detector. Version 4 will be somewhere along the lines in the future.
Okay, I'd like to introduce the next speaker very briefly. This is going to be Stephen Leavitt, who's going to talk to us about the genetics of Cladonia. Are you all good to go, Laura? Guys, we're going to get there, don't worry.
No problem.
We've got a good view of you now. You just need to share your screen.
So I'm not seeing anywhere to share my screen.
Do you hear that, Laura?
We could try this. Let me see if we could.
It should be in the bottom of in your Zoom controls in the center.
Okay, here we go.
There you go.
You got it. Tell her to do it, make it full screen. Yeah.
Yep. Got it. It looks good.
Okay, good. Thanks for engaging with me virtually. This is really exciting to share some of the work that we've been doing. So Aaron helped kind of facilitate a really cool project where we got some funding to do some sequence data for Cladonia perforata. This is the perforate reindeer lichen. And I had a bunch of students that were engaged with this project, and Aaron David and Sterling and a whole bunch of other people helped really get the permitting done, do the sample, the field work and the sampling. And we ended up with what I think is going to be a really informative perspective into this really fascinating lichen, the Cladonia, the perforate reindeer lichen. Let me see. As we think about lichens and their role in the community, I want to highlight the fact that that Cladonia perforata, this federally endangered lichen, it was first designated as an endangered species in 1963 with a subsequent revisit in 1993. But it's not the only threatened and endangered lichen. And in fact, there are a whole bunch of different lichens that occur free on the soil. We call these vagrant lichens. And I have a couple examples here. On the left, you can see the manna lichen. This is a lichen that never occurs on rocks or soil. It's totally rock or on trees, but it's always free on the soil. In the center image, we have what's called the coral lichen. This is a lichen that's found only occurring, again, free, never attached, and is close relative to the rock posea side by side with it. On the left, a tumbleweed lichen. These huge morphological shifts when it goes from occurring on rock or bark to living free on soil. and Cladonia perforata that occurs in some isolated populations in Florida is one of these fascinating lichens that's, you know, sensitive and vulnerable to all kinds of changes. So some of the great work by Brenda Herring, she recently did a status service a status update for Cladonia perforata, looking at its total distribution across the state of Florida. And we see that Cladonia perforata, this perforate reindeer lichen, occurs in what we typically think of as 4 distinct regions, in the Panhandle on Santa Rosa Island, on the Lake Wales Ridge, and further west in the Polk uplands, and then along the Atlantic coast. And these 4 main regions have typically been considered, you know, isolated, distinct populations. You can see in the image on the right these beautiful communities that are maintained by occasional fires that maintain open space and allow for these lichens to occur. And the specific image is from Santa Rosa Island by Brenda Herring. So these lichens are rare. They occur in isolated populations, distributed across a lot of the state, but only in these upland rosemary scrub communities or other scrub communities. And we started to get interested in thinking about how in the world is the genetic diversity going to help us understand and inform the way that we manage this endangered species. So currently there's maybe 50 to 60 known populations or subpopulations, and all of these populations are occurring in these 4 main regions. So by using genetic sequence data, we thought, let's take a look to better understand kind of the past or the historical processes that gave rise to its distribution. When we think about the Pleistocene climatic fluctuations and the shrinking and the extension of available terrestrial habitats, these Cladonia populations were likely impacted by that. Cladonia, as I mentioned, they occur in communities that are maintained by occasional fire. So it lives this kind of like living on the knife edge of being sensitive to fire, and fire can kill this off quickly, but also requiring the fire to maintain the appropriate habitat in these dune systems. And then in the image in the top right, we can look at the habitat where these things occur. And these uplands are often some of the earliest to be developed over time. So development, agricultural or urban development plays a huge role. Right here, I don't know if you see my pointer, but there is a teeny tiny known population that's surrounded by all kinds of urbanization. So when we think about Cladonia perforata and try to envision what's happening with the species, we can think about the fact that fire plays a critical role. There's some dispersal limitations. So how does it get around? Is it bird? Is it wind? Are they frequent dispersers, or is it more of a less frequent thing? What's the role of urbanization? How do we conserve these? How do we even introduce genetic variation in these? They are thought to reproduce only through asexual propagules. So we thought, let's use some sequence data to help tease out some critical information in this fascinating lichen. So to do this, I'm going to talk about maybe 2 methodological approaches. The first is looking at distribution modeling throughout the most recent Pleistocene glacial fluctuations, the last interglacial, and then the most recent glacial maximum. And then we'll compare those with the present models. And then I'm going to talk briefly about how we use genome scale data to start to inform dispersal capacity, the origin of the origin of genetic variation, and to test a couple of specific questions. And then we'll conclude by talking about some of the implications of this study. So to begin with, we took the known occurrences for Cladonia perforata, and here in this image, we can see those known occurrences all the way up in the population in Santa Rosa Island on the Panhandle, to the Polk uplands, to the Lake Wales Ridge, and then eastward on the Atlantic coast. And using that information and bioclimatic variables, some that were reconstructed from paleoclimatic inferences, and others with contemporary inferences, we end up being able to model using Maxent the distribution or the predicted suitable habitat for Cladonia perforata across the entire Southeast. And we see that 130,000 years ago, that Cladonia perforata, its area of highest probability of occurrence was where it occurs contemporarily, on the Lake Wales Ridge, the Atlantic coast, the Polk uplands, with a lower probability of occurrence in the Panhandle. And jumping closer to the present during the last glacial maximum, when there was much more available terrestrial habitat, again, we're seeing this consistent pattern where Cladonia perforata likely occurred, or the highest probability of occurrence was where it occurs contemporarily, in those same regions with a little bit of an increase in probability of occurrence, where Santa Rosa Island is now. And consistently, as we're looking at these models, the variable that has the best predictive power for these models is temperature seasonality. That's consistent across all the 3 different distribution models I'll show you. And now we're looking at the current or the contemporary distribution. And again, we're seeing that same pattern where these data or these models suggest that Cladonia perforata occurs where it's known to occur today, and that makes sense. So looking at this, we're seeing this evidence of that the areas with the highest occurrence probability, they've been relatively stable, at least for the past 130,000 years, and especially in the Lake Wales Ridge and the Atlantic coast and Polk uplands, and less so for Santa Rosa Island. But these habitats have been pretty stable, at least according to these models, are suitable for Cladonia perforata. And it doesn't look like Cladonia perforata was expanding and contracting and shifting range over time. So moving forward, we said, well, now let's see what kind of insight we can gain from using genetic sequence data. So here I have the distribution of our sampling sites, and we were able to collect samples throughout the entire known distribution of Cladonia perforata. We sampled, I think, 18 of the subpopulations representing nearly the entire range. And from this, we were able to use the RAD-seq, or a reduced representation sequencing approach, where we were able to generate 26,000 loci distributed across the entire genome of Cladonia perforata. And using the variation in these loci, and these loci, I have to say, they spanned almost 1/10 of the entire known genome of Cladonia perforata. That means we ended up with data spanning over 3.5 million base pairs. So this is a huge, powerful data set that we can use to help make inferences in terms of population structure for the species. So building a tree, we see that rooting this actually became pretty problematic. But most likely, the root of the tree places the populations in Santa Rosa Island as sister to all of the other known Cladonia perforata populations. And the Lake Wales Ridge and the Polk uplands were recovered within a single monophyletic clade, and that's shown in this highlighted in blue. And then the Atlantic coast populations were also recovered in a single monophyletic clade that's highlighted in yellow. So as we look at this in the Panhandle, we sampled 3 populations. There was only a single genet or a single clone in those populations. In the Southwest, there were 3 populations that we sampled, representing only a single clone. Lake Wales Ridge, we sampled 9 populations, and there were 10 distinct clones or genets. And then on the Atlantic coast, we had 3 populations and 6 different genets. So from this genetic diversity, we see strong evidence for sex happening in what we thought was a clonally reproducing organism. Somehow they're doing recombination and having sex. We have geographically structured genetic diversity, and in most cases, the genets or the clones are geographically highly restricted. The areas with the highest genetic diversity are the Atlantic coast, especially the Atlantic or the Arbor subdivision and Juniper Ridge Research Natural Area. And then the Lake Wales Ridge, of course, has high genetic diversity, particularly in the Lake Wales Ridge State Forest. Again, Santa Rosa Island has only limited genetic diversity. So what this tells us is they're dispersal limited. These things are not, the genets or the clones are not going long distances. populations in Santa Rosa Island and the Atlantic coast are likely not the result of long-distance dispersal from the most stable Lake Wales Ridge, but likely represent long-lasting genetic diversity in those areas. So what can we do in terms of conservation? That's the question. That's where I want to leave this talk hanging, is how do we better conserve this federally endangered species with the insight that we glean from Pleistocene glacial models or distribution models coupled with genetic diversity. How can we use this to improve conservation and management for this really fascinating lichen? Thank you.
Thank you, Stephen. Let's see, we've got some questions. Go ahead, Aaron.
Hi, Steve. This is Aaron. That was a great talk. So cool to see that come together. I was curious if in these analyses you'd be able to detect, say, like hybridization between units or something like that.
Yeah, can you hear me okay?
Yeah.
So we can. So with this kind of data, we can test for specific lineages or genets that are hybridizing with each other that may be, doing sex together. But it's a little more complicated than that because that sex has happened from probably multiple lineages at different times. So inferring the timing and the specific clones involved in that becomes a statistical mess. But we could potentially.
Are there other questions? Maybe I could ask a little question. I was sort of interested in the tree that you presented, and there was this sort of cluster of the Archbold genets, but they didn't seem to be closely clustered geographically. I mean, it seemed to be sort of odd combinations that were together, particularly on the Lake Wales Ridge. Was I just dreaming that, or is that actually an observation?
Yeah, so one test that we've done is what's called isolation by distance. Does geographic distance do a good job of explaining the patterns that we see? And it does not. So what you saw is correct, that it's just this weird hodgepodge of relationships of not necessarily things that occur in close proximity. So that challenges, as we think, how do they disperse? It doesn't make the sense that we would have thought if it was just through vegetative fragmentation.
Okay, well, thank you. All right, well, if there's no other questions, thank you very much, Stephen. That was a great talk. And we're going to move on next to another online person, Yuxi Guo. Hi, nice to see you, Yuxi. We're looking forward to your talk. Yuxi has been a long-term researcher with Jean Xiaoshu at Buck Island Ranch, and she's now heading up several of her own projects. And she's going to talk today about agricultural land management and grazing intensification in relation to soil microbes. So we are getting, I'll tell you when we're ready, Yuxi.
I'm ready. Can you hear me?
Yeah, and we're good to go. We can see you. Very good.
Okay,
that's
great. Thank you. So hello, everyone. First of all, thank you for having me. My name is Yuxi Guo. I'm a postdoc from the University of Florida, and I'm excited to share my research collaborating with Betsy and Zhang Xiao on how agriculture land management affects soil microbial carbon use efficiency in subtropical grasslands. Okay, as you may already know, soil organic carbon is very important. It represents the largest terrestrial carbon pool, containing roughly 3 times more carbon than the atmosphere. Beyond the carbon storage, it's fundamental for soil health and agricultural productivity, supporting crucial functions like maintaining soil structure, water retention, and nutrient cycling. And soil microbes such as bacteria, fungi, and archaea, they play significant roles in regulating the carbon cycling in ecosystems. Firstly, as decomposers, they break down complex organic compounds from plant litter, releasing nutrients and energy. But these microbes can also function as assimilators. They contribute to carbon accumulation through biomass synthesis and metabolic byproducts. In fact, microbial nicromice contribute about 50% of soil organic carbon in grassland topsoils. A critical metric for understanding these processes is microbial carbon use efficiency. This measures how efficiently microbes convert available carbon into biomass versus releasing it as carbon dioxide through respiration. Recent research has shown that CUE has about 4 times greater influence on global soil carbon storage than other factors like carbon input, decomposition rates, or vertical transport. Many potential mechanisms have been discussed. So for example, the compounds that microbes produce, such as their biomass and metabolic products, are very good at forming stable bonds with soil minerals. They create these strong organic mineral associations that help protect and preserve soil carbon for the long term. And that's why in our study, we would like to particularly focus on understanding the factors altering soil microbial carbon use efficiency. And agricultural land management represents one of the most significant drivers of global change, transforming natural or semi-natural ecosystems worldwide. For example, in grazing-dominated humid subtropical grasslands, the agricultural land management typically involve several integrated practices. So as shown here, this includes species conversion, where native vegetation is replaced with more productive forage grasses, and regular fertilization to enhance productivity, lime application to adjust soil pH, and modifications to drainage or irrigation to control the soil moisture conditions. And livestock grazing intensity is another crucial management practice that can vary significantly across the landscape. So based on this background, we developed 4 key research questions to our study. First, we wanted to understand how agricultural land management and cattle grazing intensities affect soil microbial carbon use efficiency, as well as the related carbon metabolisms like growth rate or respiration rate. And second, we want to know what changes occur in soil physiochemical properties under different management regimes. And third, we would like to identify which soil properties are the primary drivers of microbial COE. And finally, we would like to unravel the direct and indirect pathways of agriculture management intensification affect microbial carbon use efficiency. So our study was conducted at Buchan Ranch, where we had the opportunity to examine 2 levels of land management intensity that have been maintained since the 1960s. The intensively managed pastures were extensively planted with highly productive bahia grass, received regular nitrogen fertilizer plus phosphorus and potassium until 1987, and had lime applications. These pastures also featured extensive drainage systems through constructed ditches and sometimes received irrigation during dry seasons. In contrast, our semi-natural pastures represent a lower intensity approach, where they did have some bahia grass introduced, but the planting was much more sparse. These pastures never received fertilizer or lime and had only minimum drainage modifications. No irrigation was applied. And the right map showed the 8 experimental pastures we collected soil samples for this study. We implemented a nested factorial design, crossing these 2 management intensities with 2 levels of grazing pressure. The grazing treatment ranged from 50 to 273 animal unit days per acre, representing the range of the original practices. And to quantify the microbial carbon metabolisms, we incubated our soil samples for 48 hours with oxygen 18 labeled water. After the incubation, we measured how much labeled oxygen was incorporated into the microbial DNA for getting their growth rate. At the same time, we measured respiration by tracking the CO2 accumulation in the incubation container. These measurements together allow us to calculate the microbial carbon use efficiency. And in this study, we measured a comprehensive set of soil properties to understand how land management affects microbial COE. So our measurements include from very basic soil property like soil moisture, compaction, pH, and label carbon, nitrogen, and their stoichiometry ratios, and also to the base cation availabilities, including calcium, magnesium, potassium, and sodium, as well as multiple forms of iron and aluminum oxides. We organize these properties into 4 functional categories based on their potential roles. So the accessible substrates directly feed the microbes, and their stoichiometry can tell us whether the nitrogen is the limiting nutrient. And also base cations are essential for microbial growth and enzyme production. The mineral conditions can affect organic carbon production and availability and further alter microbial carbon use efficiency. So for the stats, we firstly applied multiple regressions to quantify how our 2 management treatments, land management intensity and grazing intensity, affect both microbial carbon metabolisms and soil properties. Second, we used univariate linear regressions to identify which soil properties were the key drivers. And finally, we employed structure equation modeling to examine the direct and indirect pathways through which treatments influence microbial COE. And also because many soil properties were correlated, we used the principal component analysis to integrate highly correlated variables into relative consolidated metrics before building our pathway models. And now let's look at what we found. These bar plots show us how land management intensity and cattle grazing intensity affect microbial carbon metabolisms. So in each panel, the left 2 bars represent semi-native pastures, and the right 2 bars are from intensively managed pastures. And looking from left to right, we can see the effects on microbial growth rates, respiration rates, and COE. So land management intensification significantly increased microbial growth rates by 56% and promoted overall COE by 28%. Interestingly, higher grazing intensity worked through a different way. It decreased the respiration rates by 23%, while increasing COE by 19%. And then let's look at how our management treatments altered soil properties. We firstly found that land management intensification had strong effects on soil chemical properties. Intensively managed pastures had higher soil pH, which due to the regular lime application, and higher extractable organic carbon and nitrogen, likely due to the fertilization. As for grazing effects, we observed that higher grazing intensity primarily impacted soil physical properties such as water content and compaction, which is likely from the animal trampling effects. And in addition, land management intensification also had substantial effects on the base cation availabilities, shown in the top row, and as well as the iron and aluminum oxides shown in the bottom. So in intensively managed pastures, we can see there are very high levels of exchangeable calcium and magnesiums. Yeah, and for the grazing intensity, it's pretty interesting with some interactions with land management. If we look at the bars of M and N, so there are two forms of iron oxides. We can see the grazing intensification only reduce their contents in intensively managed pastures. So through the regression analysis, we identified several key drivers of microbial CUE. The strongest relationship was with soil pH, explaining 21% of the variation in CUE. This suggests that the more neutral pH condition in managed pastures help to support efficient microbial carbon use. We also found significant positive relationships with resource availability reflected by the extracted organic carbon nitrogen. And beyond the pH and labile substrates, we also find several other significant relationships. So the exchangeable calcium, magnesium, and sodium showed positive correlations with CUE. This makes sense because they play essential roles in microbial enzyme function, membrane stability, and cellular osmotic balance. Furthermore, aluminum oxides, both total and organically complex forms, showed positive relationship with CUE. This was somewhat unexpected because we originally hypothesized that metal oxides might reduce CUE through the mineral protection of organic matter. Instead, this positive relationship might suggest that the formation of aluminum oxides is reducing the toxicity of free aluminum ions, creating a more favorable condition for microbial growth. And for this final diagram, it shows the complex network of relationships we covered. Looking at land management intensification, we find it primarily operated through indirect pathways. It improved soil conditions in multiple aspects, like increasing substrate availability, raising pH, and altering mineral conditions, which further influence the microbial carbon use efficiency. There are also interrelationships among soil properties. So for example, land management intensification enhanced the labile carbon and nitrogen through both directly and indirectly via changes in soil water content. And grazing intensity showed primarily direct effects on CUE. This direct pathway shown by the arrow at the bottom, suggesting there might be other things going on that were uncaptured through our measured soil properties. So for example, maybe grazing is changing the composition of microbes, shifting who are present or active in the system. So we will need to explore these possibilities in the future. And in conclusion, our study demonstrates that agriculture management can enhance microbial carbon use efficiency through different pathways. Both intensive land management and cattle grazing can be beneficial to soil organic carbon storage when properly managed. And soil chemical conditions, particularly pH and accessible substrates, are emerging as the key controlling factors. And these findings have important implications for our land management, suggesting that integrated approaches may be better for both agriculture production and carbon sequestration. For example, when applying nitrogen fertilizer, it would be better to combine with the liming practices. And understanding these microbial processes would definitely help us to make better management decisions. And thank you.
Okay, thank you, Yuqi. We could entertain one question while Rachel comes up to get yourself ready, Rachel. Betsy Boughton has one question. Speaker's just coming, Yuqi.
Oh, thank you. Hi, Yuxi.
Hi,
Great job. Lots of data.
thank you.
I was wondering about, you mentioned that intensification could increase the flability of carbon. So that means like it could be more easily lost from improved pastures. And so how does that relate to carbon use efficiency? Does that mean that you could have increased carbon use efficiency, but still potentially lose carbon more easily in the intensive pastures?
Good question. I think the carbon use efficiency is all about the balance. So if the microbial community, they can allocate more carbon into their biomass, like relative to their respiration, then like from the proportion of the total carbon in the system, it would get more carbon like stored in the soil.
Hey, Yuxi, we'll have to stop it there, and I'll need to ask you to stop sharing so we could get the next.
Oh, sure, for sure. Sorry.
Thank you so
How to stop sharing?
much.
No. Stop sharing. How about now?
I think you've done it. Thank you.
Cool. Sorry.
Very good. Okay, we're on to the next talk. Thanks so much, Yuxi. Yeah.
All right. My name is Rachel Fetters. I'm a research assistant in the herpetology program here at Archbold. And I am excited to share some of our telemetry tracking data on long-term space use of female gopher tortoises here at Archbold. So which one of these? There we go. Before I get going, I just want to definitely say a big thank you to everyone who's helped with this project over the years, research assistants, interns, volunteers, collaborators, and our funding sources. First of all, what is a gopher tortoise? The gopher tortoise is a long-lived fossorial reptile native to the southeastern US. Tortoise populations are threatened throughout their range by habitat loss, human-caused mortality, and disease. It's estimated that populations have declined at least 80% post-European colonization, probably more than 80%. Tortoises have state-level protections across their range, and also they're federally threatened in the western portion of their range in black here on the map. Gopher tortoises are keystone species throughout their range. They dig burrows up to 40 feet long, which provide shelter for over 350 commensal species. Several of these species, especially a lot of the invertebrates, are obligate commensals, meaning that they're not found in the absence of tortoise burrows. Another important commensal is the federally threatened eastern indigo snake, and they also get cool stuff like cotton rats and gopher frogs and all kinds of neat things. Tortoises also play a role in plant ecology by consuming and dispersing the seeds of many different species of plants. So because of this key ecosystem function, it's important for us to understand gopher tortoise spatial ecology in order to maintain healthy tortoise populations with good demographic structure, to inform our management decisions in terms of habitat restoration and prescribed burning, things like that, and sustain the dependent commensal communities and larger scale ecosystem function. So what do we know about space use already? We know that male tortoises consistently have larger utilization areas than females, and that male movements are driven primarily by mate seeking and courtship behavior, as can be seen in these trail camera images. This is a male visiting the burrow of a female. And we know that females are likely more sensitive than males to habitat quality, given the energetic demands of egg production and the fitness consequences of nest site selection. You can see all of these eggs inside the body of that female. That would take a lot of energy to produce and maintain. So our research project took place on Red Hill here at Archbold. It's located right across the 2-lane from the main entrance. One of the central features of Red Hill is this central 5-hectare field. It was converted a long time ago from sandhill to non-native forage grasses, primarily Bahia grass, with other native and non-native species as well. And I'll refer to this as ruderal habitat throughout the presentation. This ruderal field is surrounded by about 160 hectares of sandhill and scrub vegetation, showing this kind of orange and dark green color on the map. And the sandhill and scrub has a lot of palmettos, oaks, and a variety of native herbaceous plants. And herbaceous forage is a much lower density here than in the ruderal field. Red Hill is managed using prescribed fire, but some units are fire suppressed. And there are an estimated 120 adult tortoises living within the map area shown within this the entirety of this rectangle. Densities vary greatly between habitat types. In the ruderal field, there are approximately 7 to 8 tortoises per hectare, whereas in the sandhill and scrub, it's closer to 0.5 to 1.5 tortoises per hectare. And finally, in the ruderal area, juvenile growth rates and female body sizes and clutch sizes are greater on average than in the sandhill and scrub. So there are quite a few differences going on here in our different areas. Some of the overarching objectives were to look at the effects of resource availability in the form of ruderal versus sandhill habitat types and individual tortoise traits such as body size on longer-term space use of female gopher tortoises in this area. Another objective was to examine different methods of home range estimation, comparing minimum convex polygons, or MCPs, with a newer method of autocorrelated kernel density estimation, or AKDE. However, ultimately, the choice of home range estimator did not affect our results. So for the sake of time here, I'm just going to report the results for the AKDE. So what did we expect to happen? When it comes to space use, we think that both forage resource availability and size-mediated social dynamics could be important. Tortoises are not generally considered to be territorial. However, in these high-density populations, female-female aggressive interactions are fairly common. Given that this area has an uneven distribution of forage resources and high population density in forage-rich areas, we developed a couple of hypotheses. So our first hypothesis will deal with the home range size. So first, we expected home ranges containing a greater proportion of that ruderal vegetation type will be smaller than home ranges in scrub or sandhill, because the high forage availability in ruderal habitat means less space is needed to meet energetic needs. Second, we expected that larger tortoises would have smaller home ranges because large females are able to competitively occupy resource-dense and population-dense habitat, when the smaller females would kind of be excluded in these habitats. Our second set of hypotheses dealt with the stability of home ranges, so how much these tortoises are moving around over time. We expected that home ranges containing more ruderal habitat would be more stable due to the reliability of the food resource, necessitating fewer spatial shifts in response to seasonal or annual changes, for example, like a shortage of grass due to a drought or something like that. Finally, we expected that larger tortoises would have more stable home ranges, again, due to the aggressive female-female interactions, allowing larger tortoises to kind of win and keep their spot, and smaller tortoises being kicked out. Beginning in spring of 2015, we tagged 37 female tortoises. With radio transmitters, you can see an example of how they're attached with epoxy to the shell. We ended up with an average of, we tracked tortoises weekly, ending up with an average of 329 points per tortoise. I think about 12,000 observation points total. Most of the time, tortoises were located at a burrow site. You can see burrow. Tortoises were recaptured every 1 to 2 years for body measurements. It was sometimes a little bit difficult for them to come out. One female died, unfortunately, in 2017. She was removed from analyses. And we ended up with 7 full years of data from July of 2015 through June of 2022. So a pretty substantial data set. Okay, so we ended up with these 12,000 points. And here you can see an example of 3 of our females. The small black points represent those individual tracking observations. So that tortoise was tracked to these points at various times. The clusters are around burrow locations. And using this location data set, we calculated 2 main response metrics. Our cumulative space use was based on these 95% autocorrelated kernel density estimates, or AKD. I won't go into it too much in depth, but one of the main assumptions for this method is that of range residency. And that just means that these females aren't migrating, they're not dispersing long distances. They're generally staying in a range. And those are represented by the gray lines. To look at interannual stability, we calculated the mean distance between annual centroids. So each of those 7 years has an individual centroid. And then we measured the distance between pairs of years and took an average. Our explanatory variables included vegetation community, so the percent of the cumulative AKDE area that is in ruderal habitat. So here you have high proportion ruderal, medium, and low. We looked at body size, which is just the carapace length from the back. And there we go. Age is unknown for most of these animals, and age and size are not tightly correlated. However, we know that younger tortoises grow more quickly than older tortoises. Therefore, amount of growth or change in carapace length over a study period was used as kind of a rough proxy for age. To look at cumulative space use, we used linear models on our log transformed response variable and AIC model selection and averaging. To assess interannual stability, this variable, or this response variable was not normal. We couldn't really transform in any way. So Spearman's rho is a non-parametric correlation coefficient that we use to look at interannual stability versus mean carapace length and versus that proportion of ruderal habitat. Let's start with some basic, super basic results. Mean home range, that AKDE, was approximately 1.2 hectares with a fairly wide range and a fairly broad standard deviation as well. And then stability, so that interannual mean distance was about 40 meters with also a pretty broad standard deviation and a very broad range. Some of them hardly moved at all, and some of them were moving quite a bit. The differences in sample size, again, are due to that range residency assumption. We had to remove a few females that did not meet that assumption. So that's why these two sample sizes are different. Okay. These are the results of the AIC model selection. These are the two top models that came out. They both contained the proportion of ruderal habitat, and the second best model also included body size. Just a note, even though female body size and proportion of ruderal habitat were moderately correlated, our VIF test for multicollinearity indicated that it was okay to include these in the model together. And we'll look at each of these variables individually in our model averaging. Here's our model averaging. So looking at proportion of ruderal habitat, the model average estimate was negative, and the confidence interval did not overlap 0, indicating a significant effect of this variable on cumulative home range size. And for body size, the model average estimate was negative, and the CI confidence interval did overlap 0, indicating a non-significant result. Looking at the Spearman's rho results for interannual stability, there was a negative significant correlation between interannual distance on the x-axis and the cumulative proportion ruderal on the y-axis. And then there was also a significant negative correlation of mean interannual distance and body size. So where does this leave our hypotheses? As expected, home ranges with a greater proportion of ruderal habitat were smaller than those with more scrub or sandhill vegetation. We did not find a significant relationship between body size and home range size. As expected, home ranges containing more ruderal habitat were more stable over time, and larger tortoises had more stable home ranges. So one takeaway, our 7-year home ranges are comparable to 1 to 2-year home ranges reported elsewhere in the literature. So one thing that I might have expected to see was that our home ranges over a long period of time would be larger than elsewhere, but it does seem like if they're into place for 1 year, 2 years, you're going to be using that same kind of small area over a longer period of time as well. That being said, there's a lot of individual variability and a lot of variability depending on habitat. So if you want to protect 100% of your adult female tortoises, then you can't just refer back to that mean. You need to look at the whole range of sizes that different animals might need. In terms of Archbold's land management and restoration, we're learning that tortoises don't always respond directly to our restoration efforts. For example, a large female who's established in a ruderal habitat will probably not move into adjacent sandhill, even if it's restored and in great shape, and we're really proud of it. However, that female's offspring and smaller females might disperse into that reservoir. And finally, for some future directions, we're interested in looking more at our fire data and examining how tortoises might respond to our restoration efforts more in depth, as well as looking at nest site selection and fidelity, and incorporating some additional older capture records into our home range estimates. So with that, thank you very much for listening.
Rachel, we could take one question while Finn comes up to get his talk ready. So Hassa, do you have a question?
Yeah. Hi, Rachel. That was a really great talk. Did you think about dropping, like, is the movement pattern for the young females, like, they move a lot during the first couple of years and then stay once they find a new home, like, place to stay, they hang down? So if you like drop the first 2 years of big movements, they all are sort of the same home range size?
So we also have a set of juvenile and subadult movement data that we are starting to begin to look at analyzing. So that will be really interesting to see how things change. We've had some really long distance juvenile dispersal events, which have been really interesting, like multiple kilometers. Whereas once they reach adulthood, it seems like they're fairly settled. The couple of females we had that did undertake longer movements, they tended to be smaller. So they were maybe responding to those social interactions. It's hard to say.
Okay.
Thanks.
You're all set to go.
Yeah, cool.
The Predator Prey program is going to.
Yes. Okay. Hi, everybody. I'm Finn. Okay, cool. Thank you. Yeah. I'm Finn. I'm from the Predator Prey program. I'm the research assistant here at Archbold. And today we'll be talking about how our program has been using the data that we've been collecting to move towards a higher resolution view of ranchland corridors. So here's the Florida Wildlife Corridor. At its most simple, it's a mixture of the dark green already conserved lands. So those include things like the Everglades or Ocala or Station. And then there's the light green opportunity areas, which are oftentimes working landscapes. So orange groves or cattle ranches, basically anything that we would prefer to stay what it is instead of becoming highways or development. And the general idea behind this is mapping a landscape with animals in mind so that they can move throughout Florida how they would have in ways similar to how Florida was before it was developed. So I mentioned ranchland. In South Central Florida, ranchland makes up a huge chunk of the wildlife corridor and just the landscape in general. So here inside this like black polygon is yellow color, that's all ranchland. And then outside the black polygon, that light brown is also ranchland. So it's a huge percentage of the landscape here. We are fortunate and kind of uniquely positioned to answer questions about how ranchlands contribute to biodiversity and function in wildlife corridors because we have access to Buck Island, which is our 10,500 acre working cattle ranch. When we think about these ranchlands, it's really easy to just say like, okay, there are opportunity areas in the corridor. They sort of just function as better than highways or development. But in reality, there's a lot of nuance and variation between different ranches because they're all made up of all of these different microhabitats. So this is a better, more accurate view. And this is that higher resolution view I'm talking about when we're talking about these ranchlands. Going forward, I'll be talking about 4 specific habitat types at Buck Island. And we're going to try to analyze how all these habitat types are affiliated with biodiversity so we can assess Buck Island as corridor and how it's contributing to biodiversity in general, because that's kind of the goal of the Florida Wildlife Corridor. So just to introduce these, they've been kind of talked about a bunch already today. There's the improved pasture, which is homogeneous and includes a lot of introduced grass species. There's semi-native, which has a higher heterogeneity when compared to improved pasture and has a little bit more patchy and cover for animals. And then there's the wetlands and the cabbage palm and oak hammock. So what our program uses is a camera trap network that was established by Raoul Boughton in 2015. It's 44 cameras, which are represented by the points here. And it's been going since 2015, so it's been producing data this whole time. Something to note is that camera traps are built to detect mammals that are medium to large. So when I'm talking about biodiversity from here on out, I'm talking about those species. I'm not talking about birds or rodents, bugs or anything like that. So it's not like total biodiversity right now. We're just talking about mammal biodiversity. And then something else to note is that we're using the number of detections as a proxy for abundance. So it's not like every animal goes and gets its picture taken once. There's a general correlation where the more pictures you're getting, the higher the abundance. But it's not just a 1-for-1. So that's just something to remember. Cameras themselves are positioned at 4 what we call trail types. There's the no trail, such as if the camera is in a field. Then there's cameras that are along cow trails, unimproved roads, and then canals. So with all of this data, we got handed from USDA a chunk of the data that was from 2015 to 2021, and that included 6.7 million photos. So that's like 6.7 million rows of data in a sheet. From there, we removed the unidentifiable images and kind of narrow it down. Matt was talking about how each camera takes 10 pictures when there's a detection. So we want it to be that each detection is represented once. Then we consolidate it down, and then we narrowed our time frame from 2015 to 2018. So when I'm talking about our results, that's just with that 2015 to 2018 time frame. So before we do any analysis, we want to make sure that our cameras were out long enough to start doing statistics with it. So this is a species accumulation curve. On the x-axis, we have a sampling effort. In this case, it's the number of days that a camera was left out. On the y-axis, we have species richness, which is just how many different species it saw. And this is just for camera 1. So it makes sense. You put the camera out, and quickly it sees a bunch of new species. But the longer you leave it out, it's not like it's seeing a new species every day. And eventually that curve kind of plateaus. That plateau is good. It's what we're looking for because it means that we're getting an accurate picture of how many species are where that camera is. So we go ahead and do that for all 44 cameras just to make sure that they're all out there long enough. They all look roughly like they're plateauing. So it looks good. Now we started using a generalized linear mixed effects model to clump the cameras by habitat type to look at the species richness. So on the x-axis here, we just have those 4 habitat types I was talking about earlier. And then we have on the y-axis, the mean species richness. The takeaway from this is that there's not a significant difference. So no matter the habitat type, the cameras are seeing roughly the same number of species. Similarly, when you group it by trail type, the cameras are seeing roughly the same number of species. But one thing that's not being accounted for right now is how many detections of each species we're getting, because the species richness is just how many species the camera saw ever. But in reality, there's a disproportionate amount of detections for different species. So on the x-axis, we have all our species. This is for all the cameras over all 3 years. And on the y-axis, we have just the raw number of detections. And it's clear that the vast, vast majority of pictures that the cameras take are pictures of cattle, which would make sense. So this sort of necessitates a different metric, which would be a biodiversity metric like Shannon's diversity index. And to explain that, if you imagine that you have 2 cameras, right? You have the one on the left and it sees that group of species, and then there's the camera on the right and it sees the group of species on the right. They both have a species richness of 7 because there's 7 individual species types in those groups. But it's obvious that one is more biodiverse than the other, and the Shannon's diversity index takes into account this evenness so that we can get a metric that sort of quantifies diversity. That's what we'll be talking about going forward. When we use the linear regression to sort these by trail type, we didn't see any significant relationship. So that just means that along the trail types, there's no significant difference in what biodiversity the cameras are detecting. But if you do sort it by habitat type, you do see that improved pasture has significantly lower biodiversity score when compared to the hammock and semi-native pasture. This result is kind of intuitive. It makes sense that improved pasture, because it's more homogeneous and has less patchiness, might have lower biodiversity. And so we wanted to see if we could use the data that we have on cattle to link or show a correlation between cattle and biodiversity. So on the x-axis here, we just have the number of cattle detections. On the y-axis, we have that diversity index. And each point here is representing one camera over the 3 years. And there's a significant negative correlation. So at the cameras where you're seeing more cattle detections, you're also seeing a lower biodiversity index. So it would seem that where there's more cattle, there's lower biodiversity. We saw this and we're like, okay, that's interesting and encouraging. We would imagine the same trend for something like wild pigs, because it's pretty well documented that wild pigs have a negative effect on biodiversity globally. So we did plot that, and we were a little bit surprised to see this because it's showing the opposite. So the x-axis this time is just wild pig detections, and it's showing a positive trend. And this is sort of to highlight something I'll come back to in the conclusions, but it's important to not see causality here because it's just a correlation at this point. So it's just as possible that the wild pig are going where the other animals are going, and that's why you have a higher pig detection when you're having a higher biodiversity. So after we looked at cattle and pig, we went ahead and did this for all of the species we detect. So the way to read this is that on the x-axis, we have the different species. On the y-axis, we have the parameter estimate. And that's basically the degree and direction of effect. So there's that dashed line at 0. If a species range is above that dashed line, it means that it's having a positive correlation. So the more of that species we see, the higher biodiversity. So that would be like the pigs I was talking about. And then if it's below that dashed line, then that means that it's having a negative correlation. So the more of that species we see, the lower the biodiversity score. So that would be like with the cattle. And then if it crosses that dashed line at 0, that just means it's not significant. So when we take out the ones that are not significant, this is what we're left with. So all of these species have some sort of significant relationship with biodiversity in terms of what the cameras are seeing. Notably, cattle is the only one that's negative. And then another thing about the cattle ones is that it's super narrow. So if you look at like the opossum one, for instance, all of it is above 0. So at all the cameras, there's a positive correlation with the opossum number and the biodiversity. But there's a huge range because there's 44 points, 44 cameras that are informing that. With the cattle one, it's really narrow. So that would indicate that across the board at all 44 cameras, the relationship that cattle have with biodiversity is relatively consistent. So we want to circle back to looking at this in a landscape way because the Florida Wildlife Corridor is about maps. And so here's Buck Island. All the points are represented by cameras. And then where all the cameras are represented by points. Here's a heat map of the cattle detections. So the lighter colors represent where we had more cattle detections. And if you compare this to a heat map of the diversity index, you can sort of see that they mirror each other in some places. So where we see a high Shannon's diversity index, we're seeing a lower cattle count. So this is just to sort of drive home the fact that on the landscape, we're seeing these trends. There's some important things to remember, though, and I alluded to this earlier. Besides the difficulties of camera trapping as a method, it's really difficult to separate out the cattle number from the improved pasture itself, because improved pasture has a lot of qualities, including the cattle number, which is a decision that humans make. There's a lot of things like how homogeneous it is, you know, all sorts of things about what cover is there. All of those things might be factoring into biodiversity. So right now we haven't separated out cattle as the only thing that's affecting biodiversity. That's one thing that we're looking to do in the future. So with our camera trap data and then also with complementary data sets from the ranch, we'll be able to say like, okay, this improved pasture didn't have cattle for this time period, and now it does. And then we can account for seasonality and isolate what the effect of the cattle is on biodiversity. That's kind of one of our next steps. And then we're also looking to incorporate occupancy modeling, which is one of the next steps on the horizon. And then this was just with 3 years of data. It was still like 400,000 detections, which is plenty to do stuff with and requires a little bit of patience with R. But we have 10 years now at this point of data from Buck Island, which is a lot. So we're trying to get that clean so we can use it to ask these fine scale questions about habitat type cover, other things like that. With that, I would like to just acknowledge that there's a lot of people that came before me in setting all of this up. There's still a lot of people involved in it. Hopefully most of their names are here. And then I'd also like to just give a quick land acknowledgement that this all occurred on Seminole and Miccosukee land. And with that, I would love to take any questions. How long is that? Good.
I have a question. Yeah, sorry. I was just curious if the number of cattle detections was independent from your diversity metric, because don't you use the number of cattle detections to calculate the diversity, and does that matter at all with this analysis?
Because it's part of the diversity index? Yeah, that was in there, I guess. So yeah, that would probably have, there's probably a way to statistically separate that out.
Okay, cool, thanks.
Yeah.
Yeah, my question is very similar, and it's just to say, I think if you pulled out the cattle detections and then calculated Shannon diversity index on everything that was left, the conclusion you made about the influence of cattle on Shannon diversity might be more useful, more relevant, because it's swamped right now.
You're right.
Yeah, so more or less the same question.
Okay, cool. Thank you.
Do you have data on people also, right? Did you correlate?
Yeah, we do have data
presence
on people, but the thing is that the people data is not as clean because it's not been all independently verified, because all of this has to get looked at by a human and not the human data. The stuff that was labeled as human got put aside, so that still needs some work.
And does that include vehicles?
Yeah, so yeah, that's another thing about humans is that there's a lot of nuance to like what you want to define as human. And so the machine learning especially has difficulty because like we want to count a car as human, but if someone parks a car and leaves it there for a month in front of the camera, we don't want that to be a human every time. Like eventually that becomes part of the landscape. So we have to, that's the sort of stuff that we're dealing with now.